To the class 11 students, this is the part 3 of the chapter cell division, and in my previous video I have completed till the topic of anaphase. Okay, uh, so till this much we have completed, and we got to know that in this phase of mitotic cell division the two sister chromatids get separated from each other. As you can see, they are attracted towards the opposite poles and they get separated from each other. Okay, so now we come to the last phase of this mitotic cell division, which is telophase. Okay, now telophase, the word telo means end, so the last phase of the mitotic cell division is known as telophase. During this phase, the daughter nuclei are formed. As you can see, the two daughter nuclei has been formed with the chromosomes located at poles. The chromosomes are located here at the poles. And this is the last phase of karyokinesis, means the last phase of nuclear division. What are the characteristics of telophase? Nuclear membrane is developed again around the chromosomes which are found at poles. So the nuclear membrane that that started to disintegrate during the prophase, now has started to rebuild again during telophase. Nucleolus, same for nucleolus. The nucleolus which started to disappear during the prophase has started to reappear during the telophase. The chromosomes become less condensed. The chromosomes which had started to become more coiled and more condensed during this prophase stage starts to move back to its previous stage, that is, it becomes less condensed and turns into nuclear reticulum, and the spindle fiber is lost. So these are the main characteristics of telophase. Now moving on to next stage, which is cytokinesis. Okay, so cytokinesis is the process of division of cytoplasm that occurs after karyokinesis, means after the formation of two daughter nuclei. What will happen? The cytoplasm will divide into two cells, each of them containing one nucleus each. Okay, now the process of cytokinesis is different in plant cell and animal cell, and we will see how it is different. Here we can see the two methods of cytokinesis, firstly in animal cell and then in plant cell. So first we will talk about the process of cytokinesis in plant cells. Basically, the special thing that is formed in plant cells and not in animal cells is cell plate. So this is the cell plate. Okay, now what is the cell plate and how it is formed? To begin with, the process of cytokinesis in plant cells, it starts with the reformation of nucleus of the daughter cells by means of formation of the cell plate. Means the nucleus, the parts of the different parts of the nucleus started to disintegrate during prophase, and after the mitosis is over, it started to reappear during the telophase. Now the whole nucleus is reforming. Along with this reformation of the nucleus, this cell plate is also forming. Okay, and in the equatorial region, so if this is the total cell, this is the equatorial region of this cell. So as you can see, small granules are present here, and these small granules are known as phragmoplasts, which are formed from Golgi bodies. Okay, and they then accumulate, more such granules accumulate together and then form this structure known as cell plate that separates the two nuclei, this one and this one. This cell plate separates these two nuclei with their adjacent cytoplasmic mass. Thus they are then separated into two separate or two different cells with one nucleus in each. Okay, and one more thing, this cell plate will then change itself chemically and will be transformed into the middle lamella of the cell wall. So you all know that plant cells has an extra layer outside of cell membrane, which is cell wall. So the cell wall, the middle lamella, is a layer of the cell wall, and this middle lamella of the cell wall is formed from this cell plate. Okay, So we are done with the process of cytokinesis in plant cell. Now we will talk about cytokinesis in animal cells. Okay. So cytokinesis in animal cells does not involve the process of formation of cell plate. Cell plate is only formed in plant cells, and from which cell wall is formed. So in animal cell it is very simple. The process of cytokinesis is very simple. It is just it starts by means of furrowing or cleavage formation. So this is a whole cytoplasm, right? So as they start to move inside, a part of it starts to furrow inside or make a cleavage. This technique is known as cleavage formation or technique, or furrowing. And this furrow is formed at the middle of the cell between two daughter nuclei. This furrow gradually deepens. It will gradually deepen and meet the other end, and will finally divide the cytoplasm into two equal parts. Okay, so this is all about the different processes of cytokinesis in animal cells and plant cells. Next we come to the topic of meiosis. Okay, now meiosis is a little bit more complex than mitosis, and it is a process by means of which a diploid or 2N germ cell, so here so this is 2N. Okay, this diploid germ cell is divided twice, first division, second division, to form four haploid reproductive cells or gametes. So 1, 2, 3 and 4, which are haploid. Here it was 2N, and these all are single N, means haploid. Okay, in which genetic recombination takes place by crossing over. So if you can see that crossing over has taken place here, and as a result of which here and also here, so crossing over has taken place, and as a result of which genetic recombination takes place, and thus new varieties are formed. So this is the basic idea of meiosis, why meiosis only takes place in reproductive cells. And the meiosis is also known as reductional division, because in the mother cell it was 2N, and in all the daughter cells, 4 daughter cells, it is reduced to a diploid, that is N. So it is known as reductional division. Now we will go into the details, or the stepwise details of meiosis. We will discuss about the total process of meiosis. To begin with, meiosis is broadly divided into two phases, meiosis 1 and meiosis 2. So let me write here. This total 4 phases consist of meiosis 1, and these next 4 phases consist of meiosis 2. So meiosis 1 and meiosis 2 consists of separate 4 phases. Meiosis 1 consists of prophase 1, metaphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and then after that cytokinesis will take place, and then 2 separate daughter cells will be formed, which will be haploid, and then each haploid daughter cell will then again go through prophase 2, metaphase 2, anaphase 2 and telophase 2, and will again undergo cytokinesis, forming 4 haploid daughter cells. Okay, so from one diploid germ mother cell, 4 haploid daughter cells will be formed by the process of meiosis. So as I have said that meiosis consists of 2 separate phases, meiosis 1 and meiosis 2, and between meiosis 1 and meiosis 2 there is a resting phase which is known as interkinesis. In meiosis there is no interphase, it is known as interkinesis, which is a brief period of time between the first and the second meiotic division. During this phase, no synthesis of DNA, RNA, protein takes place, and there is no duplication of chromosome. So this phase is known as interkinesis, and as I've already said, interphase do not occur between meiosis 1 and meiosis 2. It occurs only before the mitotic cell division. Now we will discuss about the detailed stages, means what happens during all the stages of these meiotic divisions in the next video. Okay, thank you.